It's my great pleasure to introduce Joshua Arnbrister, who is a PhD student at the University of Arizona in my lab. And just a brief introduction, Josh got his undergraduate degree from Oregon State University. After that, he worked for, was it a vector control in Oregon?
Yep.
Yeah.
Yeah.
So he has that connection to the practitioners of the vector control that I feel like is a really central part of PacVec. Then he came to Arizona to do a master's in entomology with Dr. Casey Ernst, where he was looking at the efficacy of different kinds of training methods for community health workers and environmental health workers to see how well this group could perform larval surveillance. And not to give anything away, but it turned out they were great at learning how to do this, but they do not have any time to actually do this service. And now Josh is working on, he's switched more to the mosquito and environment side of looking at vector-borne diseases, and he's going to talk today about his work modeling dengue in Arizona. So can other people not hear?
No, we can hear you loud and clear.
Okay, good. All right. Thanks. All right. So you ready to start, Josh?
Yep. I am. So yeah, thanks for the introduction, Kathleen. So I'm Joshua Arnbrister, PhD candidate at the University of Arizona. And the project I'm going to be talking about today is some studies we've been doing on the effects of weather on dengue transmission in the Sonoran Desert. So just some quick background, possibly. There we go. What is dengue? So dengue is an arbovirus vectored by the Aedes aegypti mosquito. So there's some debate about where dengue originated from, whether that was Western Africa or Southeast Asia. But it did come from a humid tropical environment, and the mosquito did as well, with the mosquito most likely originating somewhere in sub-Saharan Africa. Since then, it has spread globally to different regions, including significantly more arid regions such as Australia, Saudi Arabia, and northern Mexico. Um, so one of the things we're interested in as dengue spreads globally is what factors limit dengue, what keeps it from going further than some of these regions. And so when we're thinking about that and we're thinking about what limits dengue, we have to think about dengue holistically, right? So we have our dengue virus, which is spread by a mosquito, as well as a human reservoir and victims of this disease. And so we have this suite of relationships that exist between the virus, the mosquito, and humans. And to limit dengue, for dengue to be limited in its range, there has to be something that interrupts one of these relationships and causes it not to work as effectively. So, one of the things that has been proposed as a limit or related to these limits of dengue spread is climate and weather. So when we're looking at our dengue virus mosquito human relationship, we're looking at how the environment, in this case, weather and climate, relates to them. And so there's been a lot of work that weather variables, namely precipitation, temperature, and humidity, are strongly linked to dengue cases. And there's a couple of biological reasons for this. With something like temperature, we see that it can affect both the dengue virus by affecting how long the virus takes to incubate inside a mosquito host. But it also affects the mosquito's ability to develop, the mosquito's longevity, and also things like its biting rate and activity. With things like precipitation and humidity, we see that these have an effect on the mosquito's longevity, on its ability to breed, and also we see relationships between humidity and precipitation itself. with different types of precipitation affecting how humid an environment is. And finally, all of this, comes through as human cases that might increase or decrease depending on how these weather variables are affecting the mosquito and dengue virus. What's important to note here, though, is that these variables are different depending on where you are. So something that is an important weather factor in Bangladesh may not apply in Saudi Arabia. There's a lot of local knowledge that needs to be addressed when we're looking at how weather affects dengue and possibly limits the spread of dengue. So, for our project, we wanted to look at weather and dengue in Hermosillo. So, Hermosillo is a city in Sonora, Mexico. It's about 4 hours south of the Arizona border. And it represents kind of roughly this northern edge of consistent dengue transmission in the Americas. So we don't see a lot of regular dengue, locally acquired dengue cases above this area. And it exists in this arid desert area that's kind of marginally suitable for Aedes aegypti to survive in. And so we're interested in this because it is this marginal environment, right? And we're wondering why there's stuff here, what allows dengue to survive here, and possibly doesn't allow it to survive somewhere further north. So when we're thinking about climate and weather in this region, the big thing to think about is how seasonal weather here is defined by the North American monsoon and relatively cold winters. So what does that mean? What do I mean by the weather is kind of defined by this? Well, when we look at average temperature and average precipitation over the course of the year in Hermosillo, we kind of see this peak in the middle of the year of both precipitation and temperature. And this is the North American monsoon. This is a period of high temperatures and high precipitation in the region that gradually falls off going into the fall and winter. Specifically, temperature falls off to much lower temperatures than you're seeing during the monsoon season. And these aren't incredibly low temperatures. As those of you reading the graph might notice, it's about 16 degrees Celsius on average in the winters here. But this is the uh, threshold kind of between 14 and 16 degrees when mosquito development and mosquito survivability is heavily affected by temperature. So when we take this seasonal data and we look at what happens on average with dengue cases in Hermosillo, we see something like this. So what we're seeing is our North American monsoon happening here in June, July, and August with our peak in temperature, our peak in precipitation. And then we have a following peak in dengue cases happening around October, followed by this gradual drop off in dengue cases as we go into the winter. So this is fairly well established as what dengue looks like in this region. It's a very seasonal event, and it seems to line up with these weather events. So what we wanted to look at in this project is basically some statistics proving this. So we wanted to look at how much does weather influence dengue incidence in Hermosillo? Can we get some sort of quantification for how important this weather influence is on seasonal dengue incidence? And furthermore, do these weather variables have stronger correlations? Do they show us something about epidemic dengue outbreaks in the region? Do they affect how much dengue is occurring at a given time in the region? So to start with, we're going to look at what does weather influence dengue incidence in Hermosillo. So for our data, we collected data from the years 2006 to 2024. And this data included weather data collected from Mexico's National Climatological Database, as well as Dr. Breles Reyes at the Universidad de Sonora. Weekly dengue cases were collected from the Servicios de Salud de Sonora, which is Sonora's public health agency. And then we also collected population data from the National Institute of Statistics and Geography. And this was important because it let us convert our dengue cases into dengue incidents per 100,000, which gives us a more robust way to look at how much dengue is changing year to year because it accounts for the changes in Eremicio's populations. So we got all this data, and then we separated it into potential weather variables that we can associate with dengue. And so when we did that, we ended up with 2,156 possible variables. So these variables include things like weekly dengue incidence. So how much dengue was occurring in weeks before whatever time period we're interested in looking at, as that's been shown to have an influence on more dengue happening. Various weather variables on a weekly scale, namely average weekly temperature, average minimum temp weekly minimum temperature, weekly maximum temperature, and then the average diurnal fluctuation in temperature over the week, which is the difference between temperature in the day and the night. We also looked at weekly precipitation. The second thing we did, because we want to see what time period these variables are influencing dengue on, is a lag time. So we did a lag time of 1 to 52 weeks. So we took every weekly weather variable and lagged it 1 week back, 2 week back, going out 52 weeks to cover the seasonal variation over a year. And then we also looked at all these variables on a moving average. So this is taking the variable for every 2, 3, or 4 weeks and averaging it for that, as well as putting it back on a leg, because this can capture some missing information about mosquito development time and dengue incubation period, as these biological processes take longer than a week to complete. And the final variable we included was COVID. So we collected data over COVID years, and this did influence the reporting of dengue incidents. So we included it as a binary variable to help address underreporting at that time. So we took all these variables and we assessed the collinearity between these variables, and found that the max, min, and average temperatures were significantly collinear, meaning that when we analyze them, we will have to stratify between those temperatures to tell the difference. And then we also created Spearman correlations of all these variables with dengue incidents to determine which ones were most relevant to our study. And our cutoff for relevance was any correlations above zero point 0.4 of an absolute, with their coefficient being an absolute value. So both negative and positive correlations were assessed in this. So when we pulled these correlations out, what we got is something that looked like this. So this is a graph of the Spearman coefficients for average temperature and precipitation, and the weeks on the bottom are lag weeks. So every week is, 2, 4, 6 weeks back from whatever time period we're interested in looking at. And what we saw was that we had this peak correlation getting above that 0.4 between weeks 8 and 16 for both precipitation and temperature. And we had a peak negative correlation specifically for temperature at weeks 34 to 42. And so these seem to be the areas where variables were of the most interest to us. So to give a context of what that kind of means in terms of our seasonal weather, if we look at a week on our seasonal weather and dengue incidents in Eremiceo, and for the purpose of this presentation, I'll mostly be looking back from the time of peak dengue incidents, which is week 43. We see that the first correlation lines up with our monsoon season, this peak in temperature and precipitation, and that our second correlation lines up with January and February, where we have this drop in temperature and this colder winter weather. So it seems like these two events really correlate well with dengue incidents in the region. So using all this criteria, we brought down our final variables to 186 possible variables. This included temperature, 118 variables in there, min, max, and average, diurnal fluctuation, precipitation, and previous incidents. And so our final model that we're attempting to make from this can contain one of each of these variables. So the previous incidence, one temperature variable of either the min, the max, or the average, as these are collinear, so we cannot include them in the same model, a diurnal fluctuation, and a precipitation variable. And it was decided to use a negative binomial regression to analyze this because of the non-normal distribution of the data and because it allows us to calculate an incidence rate ratio, which we'll get into in a little bit. So we're still in the process of running a lot of these models because there are a lot of possible models, but we have some preliminary results from them. And these are the three current models we have. So model one is our maximum temperature model, model two is our minimum temperature model, and model three is our average temperature model. And we compared them using AIC and BIC. Basically, the lower the number is, the better the model fits. And so when we're comparing these litmuses for our three models, we found that model three, our average temperature, had the lowest numbers and showed that it probably fit the data the best currently with what we're running. And this model included previous incidents, average temperature, average diurnal temperature fluctuation, cumulative precipitation, and COVID. So what did this model tell us? Well, because we were able to get an incidence rate ratio, we are able to turn that into percentages of how much a change in one of these variables influences dengue incidence. So the first variable in the model was previous dengue incidence. And what this model showed is that for every increase of one case per 100,000 in a 2-week moving average of dengue incidence at a 2-week leg resulted in a 16.2% increase in dengue incidence overall. Similarly, we had our weather data or our temperature data, and it showed that an increase in 1 degree Celsius over a 4-week moving average of weekly average temperature at a 10-week leg led to a 28% increase in dengue incidence. So to kind of visualize what that means, if we're looking at our peak dengue incidence here at week 43, it's saying that this period between weeks 31 and 33, if the temperature increases by 1 degree Celsius, we see a corresponding 28% increase in dengue incidence at week 43. We also looked at the average diurnal fluctuation over a 4-week moving average at a 7-week leg, and this was a negative relationship. So for every decrease by 1 degree Celsius in this diurnal fluctuation, we saw a 14% increase in dengue incidence. So what that means, if we look at one of our visualizations again, is that week 43, our peak dengue incidence here, this period between weeks 33 and 35, for every degree less diurnal fluctuation there was here, there was a corresponding increase in dengue cases. And as you can see also from this graph, there seems to be this overall restriction in temperature fluctuation at these weeks. And finally, we saw with precipitation that for every millimeter of precipitation increase over a 4-week average at a 9-week leg, there was a 1.8% increase in dengue. Now, this seems really small, but 1 millimeter is not a whole lot of water. So if you actually look at that as centimeters of rainfall, that's an 18% increase in dengue per centimeter of rainfall. And so yet again, visualizing that, if we look at week 43 here, this is saying that the increase in precipitation between weeks 31 and 35 here corresponds with an increase in dengue incidence. So, does weather influence dengue incidence in Hermosillo? So this seems to indicate that dengue seasonality is highly correlated with weather in Hermosillo. Increases in temperature and precipitation lead to increases in dengue incidence, and decreases in diurnal temperature fluctuation lead to increases in dengue incidence. And this matches a lot of previous research coming out of Sonora, indicating temperature drives seasonal outbreaks in the region. So what we're really showing here is that the monsoon season represents this confluence of ideal temperature, precipitation, and also a restriction in temperature fluctuation that allows dengue seasonality to occur and promotes this as the dengue season in the region. So we established what causes dengue seasonality and what weather variables are within that. So we wanted to move into our second research question here of are any of these weather variables correlated with epidemic dengue outbreaks in Hermosillo? And so the first thing we have to do with that is define what is an outbreak. So the way we define outbreaks is epidemic channel or endemic channels. So here on the right is an endemic channel for dengue and Hermosillo. And what these three lines represent are the average dengue at a given time in the region and two standard deviations above that. So we have our first line here, this green one, and this is average dengue incidence in the region. Our second line, which is one standard deviation above average dengue incidence in the region, which corresponds to a warning. Anything that's kind of above this for a couple weeks would be an above average, but not necessarily an epidemic outbreak of dengue in the region. And finally, we have two standard deviations above this average. And anything that spends a couple weeks above this would be considered an epidemic outbreak of dengue. So we make this channel and we plot our dengue cases for each year across this to see which ones qualify as epidemics. What we saw is that 2010 and 2014 qualify as epidemics. So on the right here, this is the dengue channel, endemic channel again, with the cases for 2010 mapped out over it. What we see is our second standard deviation here that indicates anything above this is an epidemic outbreak. As we can see, we spent quite a bit of time above this in 2010, indicating this was indeed an epidemic outbreak. Now, 2015 did not qualify as an epidemic, but it was an above average year, meaning that it existed somewhere here in this area between the two standard deviations. So we chose to include it in our analysis as an epidemic year. So for our analysis of this, what we did was we looked at weather data for each of these years, 2006 and 2024, and compared it to the 30-year average of previous years for changes in temperature and precipitation to see if there was anything abnormal about the epidemic years compared to the endemic years. And we compared both the full year, the winter, and the monsoon. And the reason we yet again compared the winter and monsoon is because we saw in our previous work that they seem to be correlated with changes in dengue seasonality. And we used a Wilcoxon ring sum test for these comparisons. So did we find anything indicating that weather is correlated with dengue epidemics in the region? And the answer is no. So we found a lot of variation in the weather variables between these years. And some years that were abnormal for weather were warmer or had more precipitation than average, which could have maybe led to epidemic dengue, did not have epidemic dengue. And some of the epidemic dengue years were actually colder than average or had less precipitation. So there was no real specific variables that correlate with dengue epidemic years. And I think I want to draw attention to at this point that dengue epidemics are very complex, right? So we're looking at weather patterns and climate as one of our explanations for dengue epidemics. But there's other things that could be explaining dengue epidemics in the region. One of them being viral serotype. So in order for so dengue is a strange virus in that it has 4 or 5 different serotypes. And if you get one serotype, you likely have mild flu-like symptoms. But if you get a second serotype, you are more likely to develop a severe case with severe symptoms that is then more likely to get reported. So a lot of times we see that epidemic outbreaks don't happen unless there is exposure to new serotypes of dengue virus, regardless of other factors. Similarly, human mobility and travel patterns can have a fairly large influence on dengue epidemics. Specifically here, we are in an area where dengue is marginally transmissible to begin with, and it sees a lot of immigration from areas of more consistent dengue transmission. And so this could be a larger factor in explaining what causes dengue epidemics in the region. So in conclusion, does weather significantly contributes to dengue seasonality in Hermosillo, with the North American monsoon seeming to drive dengue seasonality? And temperature and precipitation are strongly positively correlated within this with dengue incidence. However, weather does not seem to be directly tied to dengue epidemics. And we had some limitations in this analysis due to our use of only clinically confirmed dengue cases, which biases us towards people who've had second infection of dengue. But it does make the whole definition of a dengue epidemic somewhat hard to work with here. Yeah, so I'd like to acknowledge my committee members, Dr. Kathleen Walker, Dr. Casey Ernst, and Dr. Pablo Castro Reyes for their help on this, as well as Dr. Francisco Rafael Barelas for his contribution of data and the funding for this project from the U of A One Health program.
Thanks very much, Josh. I think we have time for some questions, just a couple of minutes. I see somebody would want to ask their question. No questions? All right. Well, if you have questions for Josh, you can put them in the chat. Or also, Josh, do you want to put up did you have your email in any of your slides?
I did not.
Okay. It's very easy though. It's arnbrister@arizona.edu. So thank you again. And now we're going to have our second speaker, Dr. Paulina Maldonado. Paulina, do you want to start loading up your slides while I introduce you?
Yes, I'd be happy to. I hope I can figure this out. Zoom is requesting something. Let me see. I'm not sure what are you seeing?
Yeah, we're seeing your slides, but in presenter view.
Okay, so I will have to share. Sorry about that. I'm trying to figure out which is the screen I'm supposed to share.
It's always the joy. Well, can I start introducing you while you work on that?
Yeah,
okay.
yeah. Sure.
So I'm very excited. There you go. I'm very excited to introduce my colleague, Dr. Paulina Maldonado Ruiz, who joined the Department of Entomology here at the University of Arizona almost 2 years ago. Paulina did her Bachelor's of Science and Master's of Science at the Universidad Michoacana de San Nicolás de Hidalgo. Did I get it right, Paulina?
Great, perfect.
Okay, in Michoacan. And then moved to Kansas State University to do her PhD with Dr. Yusun Park. And since she's come to the U of A, she has been very hard at work, both establishing, continuing her work on alpha-gal and some other interesting tick-related control measures, but also getting to work on our dominant vector here in Arizona, which is the brown dog tick species complex. And she's been starting working, helping tribes with their Rocky Mountain spotted fever prevention campaigns. So very excited to have her in the department. And today she's going to talk about that weird thing alpha-gal syndrome, which is kind of opening a whole new area of tick-borne medical conditions. So with that, let Paulina start.
Yeah, thanks for the introduction, Kathleen. I just want to make sure can you guys hear me okay?
I can hear you fine.
Okay, great. So I'm gonna go over a little bit about what alpha-gal syndrome is, although I'm sure that this audience is very familiar with this. So for the past, as Kathleen said, almost 2 years, we've been working on continuing some of the work that we started with alpha-gal syndrome a few years ago. So alpha-gal is carbohydrate, which is present in most mammals except for the old world monkeys and humans. And you can see the little structure here at the top right. Now, alpha-gal syndrome, which was previously known initially as red meat allergy or tick bite allergy, or had different names earlier, is now officially recognized as a syndrome just because of the complexity of symptoms that occur. But it occurs due to the increased production of IgE against this galactose alpha-1,3 galactose, or we're going to call it alpha-gal from now on. And what happens is that the individual is sensitized, and after desensitization, the individual is exposed by consuming alpha-gal containing products, and they get various symptoms, which could range from mild abdominal discomfort to urticaria or anaphylaxis. So this is known as a delayed allergic response because it takes between 2 to 6 or even up to 8 hours to notice the symptoms. And I just wanted to point out that there was a first case report of death recently, a few weeks ago, that came out in November. And what was interesting about this is that we typically don't really see indigestion cases as severe, but from this case report, it seems like there was no evidence of anaphylaxis. And most of the reports that the patient had that unfortunately passed was mainly indigestion symptoms. So that was really, really surprising to see. Now, just going back to a little bit how the alpha-gal syndrome tick connection occurred, it initially started by seeing that there was some geographic distribution associated with the cases of alpha-gal syndrome and the distribution of the Lone Star tick map, which coincidentally also resembled the Rocky Mountain spotted fever distribution in the US. So here on the right, we see the distribution of the Lone Star tick, how it has been expanding, and some of the earlier reports on alpha-gal syndrome cases. Now, this tick, as you may know, it is also associated with causing other diseases that are associated with pathogens, but also non-pathogenic diseases such as a southern tick-associated rash illness. The other thing is that while in the United States, the Lone Star tick has been the only tick that has been linked to alpha-gal syndrome, in other parts of the world, other ticks are the main culprits for this syndrome. Now, while the mechanisms are yet still poorly understood, this is what has been proposed. So initially, there is on the left here showing the allergic or the sensitization step, which occurs first when the tick bites, introducing that alpha-gal, which is later recognized by either memory cells or the NKT cells, depending if it's the carbohydrate is linked to proteins or lipids. Now, this class switch of cells between memory B cells occurs and leads to the production of the alpha-gal antibodies, which are later in the surface of the basophils and the mast cells. Now, these cells are going to be having these antibodies, and at this stage, the individual has been sensitized. Now, several weeks or months even could pass before they get an allergic reaction because of the consumption of the alpha-gal products. Now, this could be any mammalian-derived products, not only meat, but also dairy. And the individual would consume it, then it would be absorbed into the bloodstream where they would encounter these cells that are already expressing this IgE specific against alpha-gal, leading to degranulation and the release of those cytokine molecules that produce the allergic response. Now, our lab is primarily interested in what's happening on this stage in the secretion. What we previously have found, and work that I did on my postdoc, was that we know that there is alpha-gal in the salivary glands of the Lone Star ticks. Now, what we don't know is how it got there. The very initial hypothesis was that, okay, the ticks acquire it from a previous blood meal from a mammalian non-primate host, and in the same intrastadial host switch, they introduce it to a human host, right? So they feed on another animal, dog, whatever other animal might be there, and then they feed on a human, then they transfer it, leading to sensitization. The individual ingests that mammalian-derived product, and then they get their allergic response. Now, this was earlier on, so we actually tested this hypothesis in an animal mouse model. So we actually characterized the alpha-gal epitope in the tick salivary glands that have been fed on bovine blood. So this diagram here, or this graph at the top, shows the percent of total of alpha-gal glycans that are in ticks that are fed in bovine blood in 0, 2, and 5 days. And as you can see here by the bar graph, it seems like, yes, they are increasing the levels as they are feeding on this bovine blood. So we tested this in an animal mouse model. So what we did is we used this humanized mouse model that is extensively been used to test for alpha-gal. And what we used is different treatments of ticks. So we infested the mice with ticks that have been previously fed on bovine blood or unfed. Now, long story short, after waiting for the immune response to build up, and when we tested for antibody levels in the mice, this didn't seem to support our hypothesis. So we weren't able to show that the tick transfer from a bovine blood-fed tick to the mice was going to cause that response. So that doesn't mean that it's not happening, but we weren't able to prove it in our animal mouse model. So the other hypothesis was that ticks can produce alpha-gal at a baseline level or during feeding. And sure enough, what we actually did was we fed ticks in an artificial blood feeding system on bovine blood and human blood, and we did see that the ticks can actually produce alpha-gal levels, even feeding on a non-alpha-gal producing blood source, which was human blood. And we see here males and females also produce. Now, the other interesting thing is that not only that, but ticks that have been feeding on bovine blood for 5 days, so partially fed ticks, some of them were 0 in alpha-gal levels, while some of them were higher. Now, this might have explained the reason why our mouse model didn't work. But the other question is, if they're producing it, if it's something that inherently genetic factor in the ticks, then we must assume that the mechanisms for alpha-gal production are unique for Embryoma americanum, right? Because so far we know that it's the only tick that has been associated with that. Now that leads me to the main 3 kind of projects that we're working on in our lab, and I'm gonna try to go over them briefly just to give you a broad idea of what we found so far. So the first thing is, are other ticks capable of producing this alpha-gal sensitizer? And if so, are they able to induce alpha-gal syndrome? The second one is, what are those mechanisms? If they are producing it, how is it? And are they actually different from Embryoma americanum or the Lone Star tick, or is it unique to the Lone Star tick? And then lastly, we want to look into the dog hosts and how this association or correlation may occur with companion animals that have alpha-gal and the occurrence of, in the presence of ticks. Now I'm going to start with what we found so far for the other tick species. And this is just to show you a current map of the alpha-gal suspected cases in the United States. And you see here where the dense region of cases is located that corresponds with the Lone Star tick distribution. However, we see other cases showing up in north and in the west. Now, we don't know the travel history of these cases, and they might have traveled to places where there's high incidence of Lone Stars. However, there's still that question that other ticks could potentially be. And just to show you an idea of what ticks we might be interested in, we know that in the East Coast, we have Ixodes scapularis, which is primarily vector of Lyme disease, and then as well as Ixodes pacificus in the West Coast. And in the case of Rhipicephalus, the brown dog tick, well, we have it pretty much everywhere. However, we know that in particular in Arizona, it is causing a problem. One thing I should point out is that just this year, we've had reports of one case confirmed of alpha-gal syndrome from an individual that got bitten by Ixodes scapularis. And then the other interesting thing is that we have this new species that for now we're going to call Amblyomma maculatum, but it is also in Arizona, and we actually don't know what status of vector, what the vector status of this tick. So we're also interested in doing that. And the way that we do it in the lab is we actually used all of our ticks were not field collected for first analysis. We used all from the tick rearing facility, and we used an artificial feeding system that we designed in our lab, where we put the ticks in here. We used a 3D printer, and this is kind of a fancy exploded model that I want to take the opportunity to brag a little bit about that we do in the lab without the need of using animals feeding. And if you're familiar with mosquito or tick feeding, you know, it's preparing a membrane and then just having the blood and the ticks access that blood, and then the ticks are kept in high humidity and warm conditions to encourage feeding. Now, this is how the chamber looks like in the inside when there's initial feeding. I would say this partially fed female is between 3 to 4 days. And then after a few days, it gets pretty nasty and, oops, sorry about that, getting a phone call. And then we see after a few days, we get this, you know, juicy females that we can later dissect, extract the salivary glands, and do quantification. And we do this quantification by using antibodies against alpha-gal in Western blots or ELISAs. And I'm just going to bring this data back up, which were the levels that we previously found when I was doing this in my postdoc. This was already published, same colony ticks, and these were the alpha-gal levels that we see in males, females, Lone Stars, and fed on bovine and human blood. Now, when we got here, we were trying to do this, replicate the same methods again, and we didn't get the exact same results, although similar trends. Now, we don't see that the Lone Star tick has, female has high levels as we used to before when they're fed on human blood. Nonetheless, we do see that there is the presence of alpha-gal still in the salivary glands of the Lone Star tick when they feed on human blood, which lacks alpha-gal. Now, interestingly, for the other tick species that we've tested Labreer Ixodes scapularis and Rhipicephalus sanguineus, and what we see is very interesting. So in the case of Ixodes scapularis, we did this test a year ago, and all the unfed ticks had basically 0 levels. We tested more ticks this year, and now we see higher levels of alpha-gal in the unfed Ixodes scapularis, which was interesting, and we actually don't know why. The other thing is that in Rhipicephalus, We do see an increase in bovine blood-fed ticks, which we would expect if they're acquiring it from the bovine blood. But the other thing that we see is also high levels in human blood-fed ticks. Now, the other interesting pattern is that in Rhipicephalus sanguineus, the males are actually the ones that have higher levels than the females. Now, I just wanted to point out here that we want to look further into this. We're collecting more data, but we do want to look a little bit more further into the mechanisms using Rhipicephalus sanguineus to try to figure out what the mechanism of alpha-gal production is. Now, moving into that, what we know so far is that the ticks produce alpha-gal, right? It seems like it's not as simple as they're picking it up from the host. It very well may be in the case of bovine blood, but when you're feeding the ticks on a human blood that is not known to have alpha-gal, and we see it there, then safe to assume that, you know, it's being produced by the tick. Now, we believe initially it could be some genetic factor, but the other hypothesis is that potentially microorganisms may play a role in this alpha-gal production. Now, in the ticks, the galactosyltransferase gene responsible for the glycosylation of alpha-1,3 galactose is not known to be found yet. However, that doesn't mean that other galactosyltransferases are not doing that role. So I just want to show what is the direction that we're taking with this project. So we currently already have the quantification of the alpha-gal levels in Rhipicephalus and in the other ticks. But this image just shows some Rhipicephalus in the artificial feeding system, and here we have an Ixodes scapularis. But the diagram I'm showing here is how we actually quantify our alpha-gal. So the graphs that you see represent 1/4 of this half of the salivary gland. So 1/4 is quantified using Westerblot, and the other 1/4 is protein that is being saved and stored, hopefully to create a pool for using for the N-glycome and detection of glycosphingolipids. Just because we know that, you know, those are also associated to lipids, and so far we've only tested the protein aspect. Now, the other thing I want to point out is that the graph here that we have at the top for Rhipicephalus sanguineus, because the males are the only ones that show in Rhipicephalus sanguineus higher alpha-gal levels, we believe it might have some correlation with aspects regarding mating or specific organs associated to male or reproduction, including the type 4 acini in the salivary glands, which are exclusive structures that are not present in the females. And all of these results are exclusively from the salivary glands. The other thing we want to test is for transcriptomics and microbiome. So because we already have the quantified phenotype and we are saving the other salivary gland, we want to use the other salivary gland pair to see if the alpha-gal phenotype correlates with any microbiome changes or any particular genes for the production of alpha-gal. Now, on the right is just to show the comparisons of what we see with Rhipicephalus versus Amblyomma americanum at the bottom, which it's somewhat shifted. The males seem to be producing higher than the females, as different in Amblyomma americanum. So the last direction, or the last kind of project that we are aiming to address is what potential associations and correlations may they be with AGS human cases and the occurrence of dogs in the presence of ticks. Now, not only for human cases, but we're also interested in seeing what are the health implications of the tick burden in dogs. So we know that dogs produce antibodies against alpha-gal after tick bite exposure. So this was already reported. Now, yes, dogs have alpha-gal. They have endogenous alpha-gal. So theoretically, they wouldn't necessarily need to produce antibodies against a self molecule, but they do when they have tick bites. And we also know that dogs suffer from adverse food reactions that so far have not really been linked to any ectoparasites. So we wanted to address these questions. And in order to do that, what we tested is this graph just shows the comparison of the levels of alpha-gal in dog host ticks. So on the left, you see the Rhipicephalus sanguineus brown dog tick and the Amblyomma maculatum, which now we know that it is in Arizona. However, we still need to test the actual field ticks because most likely this is a new species and not really Amblyomma maculatum. Nonetheless, so on this figure, I just show you the levels of alpha-gal, and this is just the line shows the 95% distribution of the Amblyomma americanum bovine fed, and you see here just the spikes that are above that line in both in Rhipicephalus sanguineus and also on the right in Amblyomma maculatum. Now, we are collecting more data to support the Amblyomma maculatum results, but we do see that the ticks are capable of producing the alpha-gal epitope. Now, as in human AGS, the fact that they carry the alpha-gal epitope doesn't necessarily suggest that they would be sensitizers. But what we hope to do with this project with the collaboration with Kathleen Walker and veterinary experts, Victoria Sens and Jennifer Wishney, is to actually quantify those alpha-gal antibodies in dogs. So what we plan to do is draw blood from the dogs that are arriving at the shelter and conduct antibody testing. And we're going to do antibody testing not only to see if they have some food sensitizations, but also against the alpha-gal epitope. And then we're going to quantify or associate the tick burden that these dogs present and try to find any type of correlation with those antibodies. Now, the other thing that I didn't mention is that we believe that the presence of endosymbionts or pathogens in the saliva of the ticks might be another factor for enhancing the production of alpha-gal. So we also want to use this system to test the ticks for pathogens and the dogs for pathogens and see if we can potentially find any correlations of this dog tick infestation with any known adverse food reactions. So what we want to figure out is if there's not only dog health, but also trying to figure out if there might be a link between the alpha-gal production in companion animals, as we know that dogs are alpha-gal producers, and if in the co-occurrence of brown dog ticks, that could pose a higher risk for humans. Now, as I mentioned before, there's a lot of questions to answer here before we make conclusions on whether all of this is actually causing alpha-gal syndrome in humans with other tick species. But we hope to continue with that work. So the take-home message, what we have found so far is that other ticks can produce alpha-gal during feeding. We see that, we can quantify that. Now, what is the biological relevance of it is still not known. The other thing is that we actually really don't know yet what level of alpha-gal in Lone Star tick is actually capable of inducing sensitization. One big question is, and one of the reasons why we believe our mouse model didn't work early on, is because now we know that repeated exposures or repeated immunizations of the tick salivary glands actually cause enhanced production of IgE. That might be another factor. And in order to know if this findings that we're seeing are relevant for AGS, we need to move forward and do the animal model, which we hope to do soon. So that's something that we're trying to address and seeing if we can actually test. I believe that there might be a shift as well in tick population, which we would also want to address. Now, the other thing is that the blood source, or in this case, we're going to call it type of host, may play a role in the level of alpha-gal. Now, endogenous levels of alpha-gal, in the case of bovine blight, for example, they actually don't correlate with the alpha-gal levels in the salivary gland of the tick, if that makes sense. So what we see is not a one-to-one. It's not like the tick is acquiring so much amount of alpha-gal in the bovine blood, and it's the same amount that we see in the salivary glands. So it is increased. So it's much higher what we see in the salivary glands. Also, the fact that we see the high levels in human blood-fed ticks. So another thing that we need to evaluate is the blood host specific component that might be present. So in the lab, we use different donors for blood, so different individuals, and we see variation. However, even within the same tick cohort and the same blood donor, we see this large individual variation of some ticks producing 0 and some ticks producing higher, even when they have been attached for the same amount of time in the artificial feeding system. And lastly, this large individual variation, we have this hypothesis that we believe it could be a genetic factor or an endosymbiotic bacteria. However, seeing this variation within same batch of ticks, which are sibling ticks or in the same cohort, makes me think that it might be something more environmental. This is why we really want to move forward and test field ticks. So we are hoping to test field ticks of Ixodes scapularis, Ixodes pacificus, the new amblyoma species that we have here in Arizona, and also hopefully we can collect brown dog ticks from dogs that are unfed that we could bring into the lab and feed them. Because if it is more of an environmental shift, then the field ticks are going to definitely be more relevant for our findings. I just wanted to thank all our collaborators and our U of A collaborators as well, the Maldonado lab members, the amazing undergraduate students here in this picture that have been doing most of the work since I've joined, and our funding sources. And yeah, if there's time, I'm happy to answer any questions you may have. And if not, my email is right there, and I have you can feel free to scan that QR code that will take you to our website and learn a little bit more of what we are doing. Aside from alpha-gal syndrome, as Kathleen mentioned, we have other projects going around, so feel free to reach out if you have any questions. Thank you.